Thanks, Brian. Good afternoon, good evening, everyone. First, I want to welcome everybody to the 6th annual Memphis Scipreneur Challenge, no longer Memphis, final pitch event and our first ever statewide rendition of this. And for those of you who don't know my name is Ryan Hughes. I'm the director of this year's Scipreneur. And before we get started, I just want to briefly say a few words about this year's program. This is something I said last year, but it's worth repeating that these last 2 years have been pretty challenging, and Scipreneur has not been immune to those challenges. It can be tough putting together a program to get buy-in from the participants when so much of entrepreneurship is the networking, is the face-to-face interactions. But despite all these difficulties, I'm really proud of everyone who participated this year. They stuck with us throughout the entire program and really put together what I believe is going to be our most polished teams leading into this finale. So, I'm really excited that they have the opportunity tonight to showcase all the hard work and effort they've put in over these last 10 weeks. I really think they're going to impress you guys, and I can't wait to hear the final pitches. So before we kick this off, there are a couple people I need to thank. First, Erica Dillard and Rohan Isaac. They have been tremendous in supporting the Scipreneur program, helping me coordinate everything, and I'm really not sure where I'd be without their help, so thank you. Then Brian and Shelby, you guys have basically been made this entire virtual program possible. You guys have worked tirelessly putting together the curriculum, mentoring everyone in the program, and leading the classes week in and week out. Your involvement truly elevates the program, so thank you. And then a special thanks to all the mentors and entrepreneurs in the Memphis ecosystem who have given their time and expertise throughout the program, including our judges tonight, who you'll meet shortly. And then finally, a big thank you to all of our sponsors, the Tennessee Research Institutions, St. Jude, UT, CBU, Oak Ridge National Labs, Vanderbilt, University of Memphis, Epicenter, and BioTN. Really, it's their continued support that makes all of this possible. So again, thank you. Thank everyone for being here. I'm really excited about tonight. I hope you enjoy it. I think we've got a great program. So with that, Brian, go ahead.
Thanks, Ryan. Thanks again, everybody, for joining us tonight. We got a really large crowd. We pivoted into the virtual world over the last couple of years as we went through the pandemic, and it actually works out really, really well. We were hoping to kind of get a hybrid going tonight, but just didn't turn out. But we know this is a great way of doing this, and I think you're going to really enjoy the format and how we move along. This year, we had 3 great intellectual property focus areas chosen by the participants in the cohort. We had several, I think half a dozen IP submitted to us that we had to choose from. All of it was fantastic stuff. On our kickoff event, and then the first night, we had an IP parade where all the IP holders talked about their IP focus areas, and then our teams picked which ones they desired to work on. All of our participants are postdocs, grad students, even engineers that have just decided that they want to get into that biomedical space as well, which is a fantastic space to get into. Everyone that took place is more on the scientific or engineering side. So this was a new set of disciplines for them to learn on how to be an entrepreneur. I can talk, and I can safely say for my other 4 facilitators of this program and Abby, our sponsor with BioTN, that this has been a very impressive group. The participation week to week was fantastic. The effort that they put through, as an educator, not having to ask someone for their homework is fantastic. We added some other elements this year, like a 60-second elevator pitch video that they had to do halfway through, and they did a fantastic job. They're making some final updates. We didn't want to release those before they actually finished out their deck in case anything changed or informed the video. So they're going to update their videos and give me their final ones, and we'll post those to the YouTube channel that we have as well. It was some fantastic stuff. So they learned how to do the 60-second elevator pitch. We took them on a journey on how to understand product market fit, spent a lot of time in customer discovery. We had some great guest speakers come in and talk about regulatory with MRC Global. And we also had some people come in and talk about financing and funding as well. So all through this whole process, we had to come to bear a lot of different resources from the ecosystem to help these individuals go from understanding that scientific space into that entrepreneurial space. And what they learned, actually, because the scientific method is a really good repeatable method, that's the same thing you use when you're an entrepreneur, of trying to figure out a theory, build a hypothesis, test, gather your evidence, document, journal, publish, talk about it, sell, make money, sell your business, and buy an island, right? So that's what they learned is the opportunity space here. And just so you know, the reason we do this, you hear the statistic that 90% of entrepreneurs should fail and startups fail. That's just their first go around, and that's without any resources. So what we want to do is be able to provide these resources. I'm really happy to say that at least one of these teams is going to continue forward with the IP holders. The IP holders have decided to move forward with the business as well. So we will hopefully be able to announce some things here, not too distant future, that we're going to continue on with one of the teams, hoping to have some participation going on with all three of the teams tonight. So in order to do this, we're going to walk through 3 pitches. Each pitch is going to be about 7 minutes in length. Then we're going to hand it over to our 3 panel judges who are going to ask questions over a 7 or 8 minute period. We'll do that 3 times, rinse and repeat, rinse and repeat, rinse and repeat. Then the judges will deliberate, choose the winners, and then we'll make that announcement at the end. We should be in and out of here by 7:20, 7:30, and I think you're really going to enjoy this time. It's going to be pretty valuable for all of us, and we appreciate you, everyone in the audience, joining us tonight. Okay, so before we get started, I want to hand it over to each one of the panel's judges to give us who you are and just a brief bio. Doesn't have to be too long. Just tell us a little bit about yourself, your background, and then we can just move on to the pitches. So I'm going to start and put Danielle Gore on the hot seat, but I know she can handle it.
Hello, I hope you can hear me and see me. My name is Danielle Gore. I am the Director of Health Tech Innovation for Epicenter Memphis, and also direct the 0 to 510 Accelerator program here in Memphis. So I work with a lot of amazing startups that come through the Accelerator program and receive funding from Epicenter, and also try to run additional programs that increase the diversity of founders that come through the Memphis ecosystem.
Great. Thanks, Danielle. Mr. Gaines, how about you?
I'll be the first one to accidentally not unmute myself.
Won't be the only.
My name is Bill Gaines. I'm also based in Memphis. I did a master's and PhD in organic chemistry here in Memphis, and then pivoted through the research and development into strategic marketing, sales, and now business development spaces. So I have an investment and advising portfolio and own and operate 2 business support agencies here in Memphis, one that focuses on software development and marketing, and the other that does kind of back office operation support for growing businesses.
And Rohan.
Hi, everybody. I'm Rohan Isaac. I'm a Patents to Products postdoctoral fellow, a program sponsored by the Epicenter and the University of Memphis here in Memphis. I'm working on commercializing a technology related to live microscope imaging of samples. And I'm excited to be a part of this very exciting panel and listen to your wonderful pitches today. Thank you.
Very much, judges. I know you're going to see some really fantastic presentations today. Not trying to put too much pressure on everybody. They've done a fantastic job. We've been practicing over the last few weeks. And without further ado, we're going to get started. Okay, so let me just make a few adjustments here. All right, so team 1, Jennifer, you're up.
Okay, let me share my screen.
Yeah, we'll let you know when it's there. Coming up now.
Okay, let me just. Okay, if you're ready, I will begin.
Go ahead.
Okay, so we are excited to present to you today the Chronos lipid nanoparticle, the plug-and-play gene therapy delivery system. So we are at this precipice of the RNA revolution in medicine, and you've already experienced this because the COVID-19 vaccine produced by Moderna and Pfizer was an mRNA vaccine, which means you've already witnessed the promise of RNA vaccines and therapeutics. So the gene therapy market in 2021 was valued at nearly $3 billion, with a predicted growth of 20% from 2021 to 2028. And the FDA expects to see 200 new cell and gene therapy investigational new drug applications per year by 2025. So we're in a really exciting space because gene therapies can achieve long-lasting or even curative effects by correcting the underlying genetic problem. And as it turns out, a vast number of diseases, even some infections, have a genetic basis. So there's a wealth of potential in treating things like vascular diseases, cancers, neurodegenerative diseases. The list is quite expansive. In fact, many of what I've represented here are chronic diseases for which RNA therapeutics actually represent the best potential intervention. But there is a significant hurdle in translating any of these therapies to the clinic, and it's the very reason you haven't seen these on the market. It's that any gene therapy relies on a delivery system, and without it, any gene therapy will be immediately degraded in the body. So that's what we want to offer you today, a lipid nanoparticle delivery system for gene therapy payloads. We are terming ourselves the plug-and-play nanoparticle. Essentially, you pick a gene therapy, you pick your disease target, we will help you package and deliver it. And we will do it better than what exists on the market currently now. So synthesized LNPs, and these are the very ones used in the COVID-19 vaccines, produce an immune response and are toxic, and they're not stable. So we can't use these for long-term treatment. And natural LNP options are poor at packaging the gene therapy payload, and what gets in is not getting effectively delivered to the target. So all of this to say, we can't use current LNP formulations in the treatment of chronic disease. And that means there's this vast segment of the RNA therapeutics market that is currently unreachable. So in step, the Kronos LNP, this is our proprietary LNP formulation and assembly method. And we address every failing of past formulations. So this is all naturally derived. It won't produce an immune response. It's non-toxic, it's stable. It effectively packages your drug at a rate of 99%, and it effectively delivers it to the target. So we can use our LNP formulation in the treatment of chronic disease. But the applications for this are far more vast than just treating chronic disease. There are applications in terms of targeting tissue specific by making changes to certain peptides on this lipid nanoparticle. This has been established already with RNA payloads, but we could theoretically look at this with any gene therapy payload, which means the swath of diseases that we can treat and prevent is even more vast. We are already validating this in a vascular disease model, but the real advantage here is that the translatability of our delivery system across any disease for which you could develop a gene therapy.
Now, there are already a number of established companies on the market today, and they already have an established drug payload mechanism, and they already have a validated target. So some of these companies include the RNA vaccine companies, the RNA therapy companies, CAR T cell therapies, and then almost to market are some of these gene editing companies. But what all these companies are lacking are an efficient and solid drug delivery mechanism. And we believe that our formulation can really set the new precedent for a better delivery mechanism. And there are a few lipid nanoparticle drug delivery mechanisms already on the market. You may have heard about Moderna's mRNA vaccine using a lipid nanoparticle. Acuitas licensed their technology to Pfizer. But there is a key difference between our lipid nanoparticle and what our competitors have, and it has to do with our synthetic steps. We only require 2 synthetic steps, whereas Acuitas and Moderna require 5 and 7. And this is really going to reduce the costs and make it far easier to develop and manufacture our technology. So we have the potential to completely wipe out the competition and spread out to the entire market. So looking exactly at our go-to-market strategy, we are already off to a great start. We have validated vascular disease models that have come out of Dr. Mountain's lab at University of Tennessee. And we can easily expand on these vascular disease models into other disease models, models that are involved with cancer, diabetes, and heart disease. So really the next step that we are looking to move towards is to form a company and license this technology from the University of Tennessee Research Foundation. This would include leasing a lab space from University of Tennessee at Knoxville, which is in close proximity to the lab where the technology was originally conceived. And then we could start performing some optimization studies, establish our SOP, And once we have this fully business ready, optimized product, we can start to generate revenue quite quickly. And so this would include partnering with pharmaceutical companies. You saw on my market slide how many gene therapeutics companies are already out there. And then we can start to actually formulate and develop these novel drug encapsulations that are going to be catered to each pharmaceutical company. And then we'll be able to actually scale up the development of our product and start executing these licensing agreements with pharma companies, and eventually expand on our LOP and IP library. And what we're looking at, if we're able to start generating, we're looking at about $550,000 in annual licensing fees for each gene target. And this is a figure taken from about mid to large pharma companies. And really, we're attracting to many of those, and also smaller companies as well. And we also, on top of that, we're going to get 5% lifetime royalties on all the revenue. So this has potential to be quite lucrative quite quickly. So what we're asking for at this stage is $2.5 million to support 24 months of operational expenses. And this will really help the initial company get off the ground. It will enable Dr. Mountain to do a few more proof of concept studies within her lab. It will allow us to acquire the lab space at UT Knoxville. We also are looking to add a few more personnel, specifically in the business development side of things, and get our discovery and development going, and some further optimization. So this is the team that's making it happen right now. We have our founders who discovered the technology in Dr. Mountain's lab. Trey Fisher has done a lot of work to get this IP going. As I said, Dr. Mountain helped concept this idea. We have Michael McCaman, who is adding 35 years of experience in closely related fields and very similar related fields. And recently added, we have the Scipreneur Challenge consultants, who are going to really help get this concept out of the lab and into a commercial product. So thank you so much for your time. We are happy to take any questions at this point. And thanks, Scipreneur, for really teaching us a lot. We had really fun with this experience. And yeah, so we'll open the floor.
Sorry, no, Bill, I was the second one to do the mute button. I'll open it up to the panel for questions.
Yeah, I'm not sure exactly which of you to direct the question to, but I'm curious for a little bit more description around what is the competitive advantage that you have over the other existing technologies. So it looks like you're faster, you're cheaper to produce. There's obviously some element of intellectual property. Can you tell us a little bit more about the specifics?
Yeah, Jennifer, do you want to go to the competition slide if you get a chance? So there's two main parts of the IP, and that has to do with the actual formulation. So the actual lipid structure there is different, and also the way it's synthesized. The synthetics, there's a quicker synthetic step, and also we have our IP holders who would be able to expand on this a lot more details. I don't know the full science, but what I can tell you is we do have two parts of the IP. One is the one-step assembly method. So it is a much more efficient process to assemble the lipid nanoparticle. But we also have this novel structure that it enables a faster synthetic process, but it also enables a higher degradability. I don't know if are we going back to the competition slide?
Yeah.
Or are we gonna show that? Yeah, I was going to show that this higher degradability is really important as well, because this has to do with the organic nature of our LNP, and this makes the lipid nanoparticle less toxic to the cells, whereas this slow degradability causes a lot of off-target cytotoxic effects, and you can't do chronic dosing. We have some preclinical data that's showing that. So yeah.
And so does the particle automatically, does it self-assemble, or is it extruded into its form?
I believe it's self-assembled with the payload. One of the other things we want to touch on that, so all of the Acuitas, Pfizer, and Moderna lipid nanoparticles are synthetic, and this is naturally derived. It's lipid or lipids that are natural to our body. And the other aspect of this is this dual functioning peptide in these orange that you can see here. Basically, this is what gives it the ability that to encapsulate the gene therapy very effectively, which is something that other naturally derived LNPs cannot do. So we have this aspect where we can internalize, and we also have these pegylated aspects which allow us for the specific targeting, because that also helps with the cytotoxicity. But
can you give us a sense of scale on this? What are the dimensions of the interior of this space?
I cannot speak to the dimensions of the interior, but these are all under 100 nanometers, which they can pretty
in diameter.
In diameter, yes.
Okay, great.
Also, I'm just curious, since you've talked about some of the differences compared to some of the competitors, you mentioned that you've done some testing. What type of results have you gotten, or have you gotten results that definitively say that it is more efficient than the competitors, like you've just mentioned on the competitor slides? Is that correlated to data, or are those assumptions based on how the lipid is comprised?
So Dr. Mountain does have some data that's basically looking at, I believe this touches on the encapsulation efficiency, which they've gotten to very high comparatively to what you would get for a naturally derived. And again, what's the advantage of this is it's naturally derived, so it's not going to be toxic to the cell, whereas synthetics are generally very good at encapsulating the payload, but they can't be used long term. So having something that's naturally derived where you can get this good encapsulation is kind of the key parameter of this. They've also done some initial experiments basically showing that on the left that you can load with your siRNA technology and actually have an effect in your tissue. And they're doing some initial work with in vascular disease models, which we touched on, from actually ex vivo tissue from humans. And one of the initial experiments that we would do, which would be part of the ask, is to actually load our lipid nanoparticle with basically a fluorescent marker that would be transcribed when it's effectively delivered to the tissue. And so that's one of those kind of proof of concepts that you can efficiently deliver it and express your gene therapy.
Also, go ahead, Bill.
Daniel, do you have a follow-up?
Oh yeah, I was just gonna ask, just kind of going back a little bit to the testing. So it seems like there are quite a few applications and a couple of disease models or disease cases that you can tackle with this. But are there key areas that you're gonna hone in on for a go-to-market strategy, or for a first demographic or customer demographic, so to speak?
Yes, so Dr. Mountain, who Trey and her are the inventors on this patent for the LNP, her lab focuses on vascular disease. So that's a very natural area for us to focus, because her lab is already doing that work extensively. They already have access to like patient populations. So this is kind of a natural progression to work in vascular disease. And again, the real advantage of this is if we can prove in vascular disease that we have an effective model for treatment, then that would make it fairly easy to translate to other disease models. So we would want to progress with the vascular disease first, and then translate this to other potential disease therapies.
Quick question. I think that on the kind of competitive slide, then you shared that your cost of production was $200 a gram. Yeah. So what does that sell for? Like what are anticipated margins when this goes to market?
We
For
did have
the,
a
like,
licensing revenue figure. I don't, if that was indicative.
Yeah,
in the back of the slide deck or this one?
Yeah, it's a good one.
Yeah, I think our primary interest would be it'd be fairly, in terms of cost for us to produce, it's fairly cost efficient. Our initial business strategy though is actually to do LNP formulations for any type of gene therapy company that has a gene therapy and they need a specialized LNP for delivery. So we would work with them to do different things like optimization and stability tests, and then assuming those things look good, we actually hand it over to them to manufacture the LNP themselves.
and
So I guess
pay a
my answer to that. What, sorry?
And then they pay a licensing fee for the right to use that formulation.
They pay a licensing fee and the royalty fees, and then obviously they would handle, but it's lucrative to them because then it's also cheaper to produce this lipid nanoparticle for them.
Smart.
Okay, that's all the time we have for the pitch and the Q&A sessions for team one. Let's all give them a round of applause for the great job that they did. They just defended that very, very well. Okay, continuing down the hit parade, we're just going to go in order. Team two, you're up next. Let me make you co-host. All right, Deanna, you should have that now. I'll let you get your slides up and then you can tell me you're ready.
Okay, can you see the
We sure can.
PowerPoint? Okay,
Yep.
Wonderful. Good evening. My name is Diana, and I'm the CEO at Blockseed, an seed engineering biotech company. Sorry, okay. Did you know that we expect to have about more than 10 billion humans on Earth by 2035? It's insane. Feeding all these people has already become a matter of national security. The worldwide demand for food is expected to increase by 80% by 2050, which will require crop production to double in order to sustain this projection. In addition, the number of genetically different crops is decreasing, which makes them much more vulnerable to pests and diseases, and makes us become more dependent on synthetic pesticides, which have huge consequences on our health and ecosystems. The best way to tackle these problems is through improved seed engineering. Currently, the seed market represents more than $100 billion, of which 30 billion correspond to the generation of bioengineered seeds, as you already know as GMOs. And the challenge is in increasing production is pushing more and more farmers to move towards bioengineered seeds that offer better yield and are far more resistant to pests and diseases. This increase in demand is expected to surpass conventional seeds in the coming years. However, there are a few caveats with the current methods of seed engineering. For example, it currently takes 7 to 13 years to market a new variety of seeds, which considerably delays progress. It is crucial that in the near future, we're able to quickly generate new varieties of seeds, especially in the event of a catastrophe or disease that could destroy entire crops and impact the worldwide food supply. To date, there are only 11 genetically engineered crops, and this is due to the complexity and high cost of developing genetically modified seeds. It is therefore critical that we improve the efficacy of new seed development and decrease the associated costs. Last but not least, our current pesticide usage is endangering the ecosystem to a point of no return, and we need to find new solutions. To address these challenges, we have developed the Blockseed technology. In short, our technology provides a better way to produce new crop varieties with improved yield, traits, and biosecurity. Because we're using blocks and don't target genomic DNA, Blockseed can generate much cheaper seeds and 60% faster than current methods. The 4 major benefits of Blockseed for our customers are in the performance, risk and cost, catalog, and technology. And all these benefits come from the fact that we can generate a new variety of seeds in 3 years. When we look at the current market, we can see that a new seed variety costs at least $1 million for conventional crossing methods and up to $130 million for genetically modified crops. This cost is mostly due to a very long period of R&D, which we expect to decrease to 3 years, and which consequently also decreases our total costs to as low as 600K. Because our technology targets chloroplasts and not genomic DNA, no regulation exists, whereas GMOs face very strict regulations. Finally, we would be able to generate pest and disease-resistant crops without the need for pesticides. As I just mentioned, our technology targets chloroplasts rather than genomic DNA, so there is no pollen drift risk, which gives us a high standard for biosecurity. This also makes our technology much less likely to become subjected to the harsh regulations that GMOs currently face. At Blockseed, we plan to have 2 revenue models. First, we have a business-to-business segment where we want to license our technology for everyone to benefit from, as well as offering our engineering services. And second, we want to create our own seed varieties and create a business-to-consumer segment, which will generate a direct cash flow and help build our brand identity. For our seed products, we want to start with the potato, which will be the proof of concept for our technology, as they're easy to grow and to modify. Importantly, potatoes represent one of the major crops in the world, and we're actually the first vegetable to be grown in space. We have already been able to actually make larger potatoes in the lab by overexpressing the tuber protein, and we're now looking to start a field trial. And this can be expanded to any traits. So we are starting by making larger potatoes, but this can be also done for any trait we want to get. As for our timeline, we first want to start the field trial for our potatoes by the end of the year and market our business-to-business services. By early 2023, we expect to get our first B2B customer and to get the necessary authorization for our potato seeds. In 2024, we expect to start commercializing them. And by 2025, we want to start co-developing new crops with other business partners. As you can imagine, Blockseed is not just an amazing technology, there's also a brilliant team behind it. We have team members with B2B expertise in technology licensing, molecular biology, and operational skills. And we also have the experts that develop this technology in our advisory board. To achieve our goals, we are looking to raise $300,000 to finance the next 18 months. These funds will pay the salary of our team, lab expenses, field trial, and also cover B2B marketing. As you can see from our financial forecast, we expect to start making profit by 2024 with the development of our engineering services as well as the commercialization of our potato line. So the 300K that we're asking will help us finance the project until the end of 2023. With this, I'd like to thank you all so very much for your attention, and also thank you so much for the Scipreneur Challenge organizers for giving us this opportunity. And we're happy to take any questions.
Great. Thank you very much. Great presentation. I'll open it up to the panel.
Sorry, I can't tell. I was talking over somebody.
Go ahead, Bill.
Why start with the product seed instead of starting with licensing?
We're doing both at the same time. So we want to have two sources of revenue.
Okay, so in that business growth, how much of that is product versus license revenue?
I can take over. At the beginning, we expect to offer the engineering services as revenue because our potato line will be in the market only in 2024. So until 2024, we would like to offer our B2B business services to be our main source of revenue. Is why we would like to develop at the same time. And specifically, the seed product will be more a proof of concept for the B2B service side. So we can convince people to use more for license, because it's hard to convince people to use a license if you don't have any product. So is why we would like to develop at the same time.
Thanks.
You talked a little bit about how you can fairly easily change some of these properties of the seeds, like their potential yield and their disease resistance. Is it as straightforward. I guess, can you maybe talk a little bit about what of those properties is the most important in the market right now, and maybe how difficult it is potentially to achieve some of those goals? Like, are there limits in terms of like how large you can make a potato, or how disease resistant you can make it?
Yes, that's a very good question. So one of the main problems with GMOs right now is that people try to integrate genes that sometimes don't even come from the same species into the genomic DNA of the plants they want to alter. And that's very laborious and time consuming, because you have to make sure that you're inserting the gene in the right place and that it's not having off-target effects. And so the screening is very slow, whereas our technology allows to insert plasmids, which is like a circular DNA, into chloroplasts. So it doesn't integrate any of the DNA from the plant. And so we can actually perform large scale screens and find the traits that, for example, increase our seeds protection from certain, I don't know, pests, or that lead to bigger potato production or crop yield. We can screen for many different things and get results really fast.
I would like. Go ahead. I would like just to add to, we can also overexpress one compound that has a high value for biopharma. For example, for cannabis, we can overexpress the cannabinoid protein, and so we can have a better concentration of cannabinoid for this kind of high value compound. But as Diana said, we can overexpress easily any protein and do a big screen and identify easily which seed can fit the market.
So who will that initial target market be for the new amazing potato?
Can you repeat your question, please?
Yeah, when you spend the next 18, 24 months building this new amazing potato, who is that potato going to be sold to? Who's that first market?
Okay, I understand. The aim to produce big potato is more asked by chips and French fries companies that are the biggest market in the potatoes, because they need big potatoes in order to make better French fries and chips. So her aim is really first to work with some farmers and prove that they can get better yield, but it's up to the farmers to sell to anyone. But our target will be more farmers that would like to work with us and increase their yield on potatoes production.
Thanks.
Also, I have a general question. You may have had a slide on this, but can you further specify, even from an IP perspective, how you truly differ from a standard GMO? In my mind, you're still modifying. So what makes you different than a GMO product, and what makes you not currently at least fall under the regulations that go with being a GMO product?
Yes, so that's an excellent question, and it's super important to make it clear. So a GMO genetically alters a plant. What we're doing is to add DNA that will not be transmitted to the next generations. So the main problem of GMOs and why they're so heavily regulated is that if you have a genetically modified crop, it can produce pollen and produce seeds that can end up anywhere and can actually mix with nature, and you can end up with GMOs in nature. And that's really problematic. And so what our technology does is that we're able to modify our seeds to make the same as GMOs, but we're not affecting nature. So this is why we need to emphasize how good this is for biosecurity.
And so we have in our vector that we call block seed, expression biomarker that makes it to express only in chloroplast, not in all the plant.
Right.
So which helps to have the biosecurity, very important. So it will not go in the pollen because it's only in the chloroplast, not in the nuclear genome, which is the most important part.
That's a good distinction. Thank you.
And have you been having conversations with farmers or other seed manufacturers to start to evaluate the market?
We didn't have the opportunity, but yes, it's a way to, we would like to talk with some company and see with them, because what we would like is to have B2B businesses, and we would like to develop a project together, because for example, this buyer that is very good in maize. So we would like to focus on other crops, and we don't want to develop by ourselves, but develop with them. And we, with our potato line, I think we can convince them more easily than if we go and just say, hey, we have just a license. We didn't produce anything, but you work with us. So we would like to have something in our hand that we can show them as proof of concept and tell them that it works, and let's work together.
Great.
Okay, team 2, great job. Let's give them a round of applause. I'm on mute. Is everyone on mute? Everybody's on mute. So come off mute real quick and give them a round of applause. It was a silent mute.
Thank you.
A silent golf. Yeah, there we go. There we go. Great job, team 2. Great job. Okay. So Madison, I'm coming to you next with team 3. I can find you in the list. All right, making you co-host now, Madison. You can get it set up. We'll check you, see, make sure we're seeing everything, and then you can take it from there.
Okay, it still says disabled for me.
Okay. Let me do one thing first. This is our only technical difficulty tonight. We're doing great. All right, try it now. Okay.
There we go. Thank you.
Yeah.
Give me 1 second. Okay. Okay.
Okay.
Oh, sorry. 1 second.
All right.
Okay. Okay. Okay.
Sorry, when you're
Hi.
ready.
So my name is Madison Sluter, and I am a PhD candidate at UTHSC. And today I'll be discussing a revolutionary anti-seizure compound. So it is said that in the US, nearly 1 in every 26 people will develop epilepsy in their lifetime, and that nearly 65 million people worldwide suffer from epilepsy. That means that it's likely that somebody you know has or could develop epilepsy. I even had a personal experience with one of my swim students experiencing a seizure during one of our lessons. So I think it's really important to talk about what epilepsy is. So let's discuss, what is it? Epilepsy is a chronic neurological condition characterized by recurrent seizures. It can affect people of all ages and has a wide variety of causes, from infections and head injuries to genetic causes, and in some cases, the cause can even remain elusive. And so even after decades of research, nearly 1 million Americans taking their anti-seizure medications still experience seizure. So our problem is drug-resistant epilepsy, which is defined as seizures that persist despite appropriate treatment. So nearly 1/3 of epilepsy patients suffer from drug-resistant epilepsy. That means nearly 20 million people worldwide and 1 million people in the US suffer from resistant epilepsy. And in the last 20 years, there has been no significant progress made to decrease the number of patients suffering from drug-resistant epilepsy. And untreated or uncontrolled epilepsy can lead to seizure-related injuries, whether that be falling and breaking something, or getting into a car accident, or even having a seizure during one of your swim lessons, which coincides with then increased hospital visits for those patients. And so epilepsy oftentimes occurs with other cognitive disorders like anxiety and depression, or and if your epilepsy is uncontrolled and you have increased levels of anxiety, you're going to be even more anxious because you don't know when your next seizure will occur. And so for the current treatment options, although they are numerous, as evidenced by the first, second, and third generation of anti-epileptic drugs, most of these drugs display redundancy in trying to suppress seizures, and so they present limited treatment options for patients. And so on top of this, many of the anti-seizure drugs are also accompanied by adverse effects, so including but not limited to tremors, hepatotoxicity, weight fluctuations, and cognitive disturbances, severe behavioral changes such as aggression, and then also renal complications. So this further emphasized the insufficient and inadequate treatment options for drug-resistant epilepsy patients. And so our solution is our novel anti-seizure drug, JW65, which targets a previously unexplored therapeutic option in the treatment of epilepsy. So JW65 offers a novel target with high target selectivity, long-lasting therapeutic effects, good brain penetration, potent anti-seizure activity, and less adverse effects than other anti-seizure compounds, all of which synergistically combine to make a revolutionary treatment option for drug-resistant epilepsy patients. So compared to our competitors, JW65 offers a higher therapeutic window and a longer plasma half-life than 5 other common anti-seizure medications, ranging from 3 generations of drugs. This means that JW65 will have longer lasting effects and will provide more dosing options for patients when compared to others. So in December 2021, we were granted a $1.15 million grant to further develop JW65 and other compounds. And then we have also published 2 peer-reviewed papers regarding JW65 that detail its potent anti-seizure activity and also the limited adverse effects. So going forward, we plan to apply for SBIR grants to further de-risk JW65 and perform specialized studies that would allow us for investigational new drug clearance. And then once we've obtained IND clearance, we plan to partner with pharmaceutical companies in the anti-seizure drug field that we would then license JW65 and have them conduct the clinical trials related to JW65. So here's going to be a schematic of what our go-to-market strategy and business model may look like when we are finally able to go to market. So our plan is to use money from investors to acquire more evidence to support JW65 and obtain IND clearance. Once there, we will then license out JW65 to pharmaceutical companies who will conduct those clinical trials. From there, pharmaceutical companies that we partner with will then sell the approved drug to pharmacies who will then distribute them to patients. Insurance companies will play a large part in the role of the distribution of JW65 as they will set price limits for manufacturers and pharmacies, but also they influence what medications are going to be covered for their patients. So following insurance companies, doctors will also play a key role as they are going to be the people prescribing our medication. And so sales representatives from pharmaceutical companies engaging with doctors to discuss JW65 is going to be critical in establishing our compound in the market. And so finally, of course, there's going to be governmental regulation at various stages of both development and distribution that will be incurred. So now let's discuss our potential for revenue. So on the left, we have figure 1 that shows how much a person with epilepsy spends annually and for what percentages each thing are spent on. And figure 2 shows that the average amount insurance companies will spend on people with either stable or uncontrolled epilepsy. So based on these 2 figures, we were able to calculate the total US market size to be approximately $50 billion. And so this takes the total number of epilepsy patients, which is 3.4 million patients in the US, times the total cost insurance companies spend on stable patients. However, for drug-resistant epilepsy patients or patients with uncontrolled epilepsy, insurance companies spend a nearly $10,000 additional dollars per year per person on these patients. And so when we consider how much insurance companies are going to be willing to spend on JW65, if it is less than $10,000 per patient per year, we would likely be able to save them money, making them more likely to cover our product. And so therefore, if we tap into a piece of the nearly 10 billion extra dollars insurance companies are going to be spending on drug-resistant epilepsy patients, we will be in pretty good standings. And so our ask is for $250,000, which will cover approximately 1 year of research. And so this ask will allow us to help reach our goals of generating enough data to apply for these SBIR grants, and then also to obtain and file for investigational new drug clearance. And so with that, I just want to thank everybody on the Scipreneur team, also Dr. Wei Li and Dr. Lexiao Li, for their help in putting this presentation together, and then the National NINDS for funding the research related to this project. And so with that, we will take questions.
Great job. Panel?
I'd love to go back to the revenue slide if we can.
Sure.
Okay, so this potential earnings calculation is done based on the total sale price of the treatment, correct?
What, can you
Right.
repeat that question? I'm sorry.
So your $9.3 billion comes from basically $10,000 per patient
Times 1 million patients, yes.
times 1 million patients.
Right.
But that's how much money goes to the pharmaceutical companies to buy the drug. How much of that comes back to the IP holder in the form of royalties?
Yeah, so that's a really great question. So it would really depend on how much we license out our company for, or our product for, to the pharmaceutical companies, and then how much they get back from insurance companies would be a percentage of that. So that is the total market that we're able to tap into. Of course, we're only going to be getting a smaller piece, but even if we tap into a small piece of $10 billion, that's still going to generate a pretty decent amount of revenue.
Sure. Do you know what the ballpark royalty fee, like percentage range is in this market?
Honestly, I don't know.
Yeah.
Lee, if you can comment on that at all. I don't know if you know that answer.
I think this is undiluted funding. So I still believe that the IP holder can earn the part that they deserve that. So yeah, that's it.
I didn't get that last part. Would you repeat that?
Yeah, the point is, so the finding we have got for this product is I think it's undiluted funding. So I think the rights of the IT folder will be protected by this mode. So you don't have to worry about that. The new the new joiner will dilute the funding, so will dilute the benefits that IP holder has. So, but yeah, sure, we don't know that which part, how much like the whatever. Oh, this is the like potential earnings.
Mm-hmm. And have you had any preliminary? Oh, I'm sorry. Have you had any preliminary conversations with pharmaceutical companies to gauge their interest in a new anti-epileptic?
Yeah, so honestly, we are really in the R&D phase still. We're not at the point where we're really talking to pharmaceutical companies. So we are really looking for money to further our development in JW65. We've recently had some really great preliminary data in animal trials when compared to Penetoin, which is one of the most commonly prescribed anti-epileptic drugs. We saw better seizure suppression and when compared to that drug using JW65. So there is still there's still a lot of potential for development. This is in the really beginning stages. So we're not really at the stages where we're talking to pharmaceutical companies. So, yeah.
I had a quick question. So it seems like a lot of these issues are very sort of long-term and chronic. And I imagine when you're doing these tests in the lab, there are some ways to sort of accelerate that time frame. But do you think there would be impact when you try to take this to market in terms of trying to do some of those R&D studies on patients in the sense that like those timescales will get much longer than sort of the timescales that you're projecting?
Yeah, for sure. So going back to the different generations of drugs, each drug kind of improves upon the last drug, but you're always going to have different side effects that pop up. Of course, you try and fix one thing and something else happens. And so just generally, when you're looking at a drug timescale, you always go back to like the initial, for our instance, like first generation drugs. Yes, they have all these bad side effects, but they also have the most data generated on them. And so Phenytoin, which is one of the first generation drugs, is still one of the most prescribed drugs despite its side effects, solely because it's got all of that information generated from it. So of course you're face those things, any sort of like long-term study, especially jumping from animals to humans, you have a big hurdle to jump over. You're going to have different side effects. You're going to have different like long-term effects and things like that. But that's kind of always a risk you're taking in that drug development field, and you try and lessen that as much as possible.
Thank you.
Also, I just had a general question about the competition. I know you spoke generally about some of the benefits of JW65, but are there, can you note some specific differences in your product specifically for those drug-resistant epilepsy patients, and how yours is better than what's currently out there?
Yeah, so currently what's out there, a lot of anti-epileptic drugs, despite regardless of the generation, typically target ion channels. And so you're either activating the inhibitory channels, or you're suppressing channels that excite your neurons. And so while that's an okay mechanism, many of the drugs target very similar like targets. Those are the two main targets. You're either trying to heighten the inhibitory, or you're suppressing the excitatory. Whereas our compound targets a fully different target, and it's actually more related to calcium signaling, which can provide an alternative target to either activating or suppressing. It's a different mechanism of action. Does that answer your question?
It does. And then again, out of curiosity, you mentioned that you're sort of in the R&D stage. Have you started to do testing, and have you started to see positive trending of data that supports that claim, since it is using something different than what's currently out there and what's sort of the standard for these types of drugs?
Yeah, absolutely. So we've recently published a paper, I believe it was actually the beginning of this year, regarding JW65, comparing it in an animal model to the most commonly prescribed anti-epileptic drug, Phenytoin. And compared to Phenytoin, JW65 was actually better at suppressing seizures. And so, yes, we have had promising animal model sort of studies.
Thank you.
Okay, let's give a round of applause. Again, everybody come off mute real quick for team 3. They did a fantastic job. That might be the one thing that doesn't work great with Zoom. All right, now everybody go back on mute. All right, thanks, Madison. I'm gonna take the co-host away now. Okay. All right, so what we're gonna do now is I'm gonna spend about 3 seconds building a breakout room for our judges. And let me just do that real quick if you ever bear with me. But while I'm doing that, I'm going to hand it over to Abby Trotter. Abby is one of our sponsors, our main sponsor, our big time sponsor, the one who really helps us get this thing off the ground every single year. So I'm going to hand it over to her, and she's going to introduce her organization and all the things they can do for life sciences companies across the state.
Thank you so much, Brian, and really fabulous job. Can everybody hear me?
Yes.
Nod your heads, maybe. Okay, great. Really fabulous job to all the teams tonight, and some great questions by the judges as well. You know, it's always impressive to attend a Scipreneur competition, whether they're in person, and I hope one day they can be again, or whether they're virtual. It is always it's always super impressive to see these presentations, and I can tell you oftentimes they're as good as any pitch presentations that I have seen. So really great job again tonight, and it's amazing to think that this kind of work is coming out of our academic institutions. Brian and Shelby and all the mentors that helped these teams this year have really done a fabulous job. The dedication that they put to this is really terrific. But I'm Abby Trotter. I am the executive director of Bio10, which is a nonprofit organization that focuses on entrepreneurship development and workforce development. And really the Scipreneur competition fits perfectly into both. I mean, we want to make sure that our graduate students and our postdocs from across the state of Tennessee are an understanding of industry and the things that are important in industry. And so we consider this a workforce development opportunity, but we also consider this a great innovation opportunity and entrepreneurship development opportunity, because oftentimes these technologies go on to, as it was discussed in the last presentation, go on to apply for SBIR funds. They go on, to receive capital. I have had the benefit over the last several months actually to be the interim CEO of an entrepreneurship support organization called Launch Tennessee. And we offer a lot of different programs within Launch Tennessee to help startups across the state. We offer matching programs for SBIR, matching funds for companies that have received SBIR and STTR funding from the federal government. And we offer direct investment in startups, seed money, and Series A money in direct investment into startups. The other thing that Launch Tennessee does is they support a robust mentor network that is engaged in the life sciences across the state. And Stephen King is on with us tonight. He's given us a wave. He is the executive director of Bio10's Mentor Network program. So for all of you that are interested in one day taking technologies into commercialization, you should certainly keep Bio10 in mind, because we can help you with our mentor network as well to develop your companies and hopefully find funding. And I just want to say thanks to Launch Tennessee and the other sponsors tonight for contributing some of the prize money that Ryan will be handing out later, and just glad to be here. So thanks, Brian. I'll turn it back over to you.
Oh, appreciate that. Everybody hear me? All right, so I'm going to tap dance for about 5 or 6 more minutes, not literally, because that would be very, very, well, it might be too fun to see for y'all to handle. So the judges are out deliberating right now. Ryan and Shelby are help working that. They'll get back fairly quickly with us. I wanted to kind of open it up to questions. I mean, this is a program that we do and have done for several years now. We have always strived to work with all the IP holders as well as the entities they work in to help them commercialize. And this is a program that we love to gather support around. So you've seen what we do. You've seen our process. If you have any questions, this is a good time to ask while we're waiting for the judges to come back. So I'll open it up.
So Brian, Lisa Jennings here. I just, I've been on these panels and I think perhaps even judged a number. We are provided, with contact information for these groups, correct? Because if there's a particular technology that's of interest to anyone viewing the presentations, do we have a way of contacting them?
Yeah, we can follow up with that. Ryan and Eric are probably the best points of contact for this area. So if any of these technologies, piqued your interest, we can definitely hook you up with the original IP holders as well as the team that worked on this. Abby also kind of represents the entire state, so she can hook up to the other entities on the other side of the state from us, which everything's on the other side of the state from us. So yeah, between the 2 or 3 of us, we can definitely hook you up.
Great.
And the opposite is also true. Like all these entities that we work with, I know Lisa, you work a lot with what you're doing in your own space, but also everyone else. We would like nothing more than to have 40 people participating in this every year, postdocs and graduates and even other students from other disciplines or even other researchers that want to kind of do the business. But we would also love to have 10, 15 different pieces of IP to choose from every year as well. I think what you can see through these programs is there is a way to teach how to be an entrepreneur, right? There is a way for these teams to take the intelligence that they already have and apply some similar skills that they've already learned and learn how to apply that to research a market, right? Because we're talking about the same thing. The lab just happens to be in the wild. So, you know, and then obviously there's some deep type process stuff related to FDA and other regulatory, but we also have resources for that as well. The city and the state, Tennessee, has no shortage of resources for both entrepreneurship and life sciences and health technologies. And you can see that represented from all of the 25 Health that's opening up in Nashville, all the other programs and venture studios around healthcare, the Nova Fund focusing on Black founders of healthcare products. I mean, these are great things coming up in our great state.
And I would even add, because everyone that's in this space knows the challenges, knows how hard this is. If you reach out to anyone in this space, particularly in the state of Tennessee that I have seen, you'll get a response. You know, help me figure this out. If you reach out to, you know, Lisa, you've always been great to help out through the years. You know, many people. So, you know, don't hesitate to reach out to Biotin, to Stephen, to Lisa, to any of the mentors or people. Tom Ballard in Knoxville is always a great connector to help. And he's on this Zoom as well. He's always a great connector to help startups. So it's a tight knit, great, open. I should say it's a strong community. It's very open to bringing people in from across the state.
Abby, I would just add, I was most impressed with the presentation. So I don't know who will win. That's up to the judges, but I think everybody was a winner tonight. I think it was great. You know, I serve on the Launch Tennessee board and the Life Science Tennessee board. So, and it's good to see Lisa Jennings again, because I go back to my UT days, Lisa, which was more than 18 years ago. It's hard to believe. Good to see
That's
you
right.
again.
It's great to connect. Yeah. Well, it is exciting. I think there's so much out there, and, you know, startups need a boost, so to speak. And then also just, you know, there are some of us who've kind of made it to the other side. So it's important to try to help the new folks coming in. And I'm particularly interested in drug development. So, you know, Tennessee could really benefit from having more biotech, more biopharma in the state. That's for sure.
Yeah, absolutely. Okay, well, as you can see, our judges have popped back into the main room here. So I'm going to hand it over to Ryan to announce our winners.
All right, thanks, Brian. First of all, I just want to say all three teams did a fantastic job. I think this is the closest the deliberation's ever been. I mean, we're literally separating the teams by a point. So honestly, you guys all are winners. I know that's cliche to say, but I honestly think you guys all did fantastic. I think usually Erica left us. She had to go somewhere tonight, but usually she makes some sort of like PowerPoint slide that has a drum roll on it. But since we don't have that, if everyone could come off mute and maybe do a little drum roll, and I can start with the third place winner of tonight's 6th annual Scipreneur competition. So please, if you can, come off mute. In third place, we have Team Blockseed. Congratulations.
Great job.
Fantastic job. And like I said, there's such a slim margin between the teams that you guys could have all been first place, depending on if we rescored it. All right, drum roll. In second place, we have Kronos LNP. Congratulations.
Good job. And then Ryan.
And now I don't know if I need to do a drum roll, but in first place, I don't know if you guys actually announced your team name, but you need to go ahead and
Tell us.
What's that?
Tell us now.
Oh gosh, I don't know that we had a team name. Just
I think JW65 is what we're going to have to go with. Product name it is.
You don't want to lose a point right now. I'd affect that.
Yeah, no. We'll just go with the product name. JW65. I think JW65 sounds pretty flashy to me. I can make anything look elegant like that. Come on. All right. Congratulations. JW65 winning first place. Woo! That's great. Thanks, Ryan.
Absolutely.
All right. So just to close out real quickly, everybody did a fantastic job. This was another year of just really a fun time for everybody. And I really appreciate everyone's participation. It's been fantastic. A lot of hard work. We're going to be posting a lot of stuff up on the YouTube channel here pretty soon. So go check that out, and we'll follow up with some more information. And we'll be back next year, or maybe even more programs, maybe even a longer program. Hopefully we'll have a lot more IP and we can do this even bigger next year. So thanks everybody for joining us tonight. You'll have a great time, great week, great summer, great everything. All right. Bye.
Great job, all. Thank you. Thanks, Brian. Bye.
Great job, everyone.